Hello everybody, this is Jason from STEM Nerds. Thank you so much for being here. In today's video, we're going to be talking about cancer, and specifically, we're going to be talking about the molecular basis behind cancer, all the things that have to go wrong inside of a cell in order for a cancer to form, and we're going to be looking at how to prevent cancer. So there are a few different types of cancers. One, there are benign cancers. These are tumors that tend to stay in one location. And then there are metastatic cancers that will spread from one part of the body to the other. When a cancer spreads from one part of the body to another, the cells have to ignore something called anchorage dependence. Most of the cells in the human body, they are confined to a single area, with the exception of cells that are going to be in the blood and reproductive cells. But most of the rest of the cells, they are either attached to one another or they are attached to extracellular matrix. And when they break that connection, usually there are internal signals inside the cell that says, hey, you're no longer in the location you need to be in, and that creates a process called apoptosis, or programmed cell death. Another thing that happens as we look at tumors is a cell has to ignore something called density-dependent inhibition. Normally, cells are going to spread out in a tissue culture plate. They're going to be a nice single layer. Cancerous cells, however, they tend to form big, massive tumors. In other words, they start to get really crowded, and that happens inside the body as well. The body does a really good job of regulating how many cells are in a certain area. Well, these tumor cells have to ignore the feedback mechanisms that tell them they can only grow to a certain number, and again, that is called density-dependent inhibition. When they lose that ability, they begin to, crowd other cells out of the area. Now, of course, for cancers to form, lots of things have to go wrong, one of which is usually an accumulation of genetic mutations. Two really important genetic mutations, one of which being a gene that codes for the protein p53. And p53 is involved in tumor suppression. It can pause the cell cycle, and it can activate DNA repair mechanisms. It can also trigger apoptosis, and so when this gene is mutated, the cell loses all of those safety mechanisms. A second really important gene is this one here. It's the BRCA, otherwise known as BRCA. There are a couple different variants, but this gene, just like the p53, is involved in suppressing tumors. It does this by initiating DNA repair mechanisms. It also helps signal when there is DNA damage. In fact, this BRCA gene is so important that when people realize they have a mutation in either one of these genes, often females will have both their breasts and ovaries removed because mutations in this gene leads to a high likelihood of cancers in those two areas later in life. One of the things that cancerous cells have to do is ignore checkpoints within the cell cycle. We've talked about the cell cycle before, but basically what happens is a cell is going to go through interphase and the mitotic phase when making another cell. And at each point, there is a critical checkpoint to make sure the cell is going through the division process properly. And you can see that here in this video, the G1, G2, and M phase checkpoints. A cancerous cell is usually going to ignore one or more of these checkpoints and continue to replicate even though it shouldn't be. Cancerous cells are also going to need a lot more blood flow because we have a lot more cells growing in an area they shouldn't be growing. And this process is called angiogenesis. Cancerous cells have to send signals out to the rest of the body saying, hey, we need more blood vessels to give us more food, to get rid of waste, to get more oxygen, to get rid of CO2. Now, it's interesting because anti-cancer drugs have actually done one of two things. There are some drugs on the market that seek to discourage angiogenesis in the hopes of starving the tumor. Unfortunately, what that does is when you're delivering other chemotherapeutics, if there's no blood vessels there, those chemotherapeutics can't get to the tumor. And so there are other types of cancer drugs that actually enhance angiogenesis to get more of the other types of chemotherapeutic drugs to the tumor to kill off those cells. The immune system is a very important part of cancer suppression. There's no telling how many times each one of us have had cancerous cells in our body, but the immune system does what it's supposed to do, and that is it recognizes when cells become unusual. And they do that because cells will send out little protein signals on their surface that tells the immune system whether or not they're healthy or sick. A lot of times this could be due to a viral infection, but sometimes it's due to the fact that the cells are starting to accumulate mutations in their genetic code and producing some unusual proteins. The immune system then recognizes those unusual proteins and kills the cell before it has a chance to make more mutated cells. This is an area of research that is under intense investigation, how to train the immune system to recognize cancerous cells before they form tumors. Now, there can be a lot of reasons why cancers form. Perhaps somebody is exposed to a carcinogen. That could be through their diet. That could be through the water that they drink, the air that they breathe. It could be due to too much UV exposure. All of these things can potentially mutate DNA. Scientists are really starting to look at diet as a potential cancer-causing agent. In the last 50 to 60 years, processed foods have become ubiquitous in our diet. It's everywhere. High sugar diets filled with processed foods have been shown to potentially lead to more cancer. And what's interesting is scientists are starting to really appreciate the mitochondria, those little energy ATP making factories inside of our cells. Turns out mitochondria has its own DNA. It's got its own ribosomes. It's basically like a little tiny machine that is almost completely self-sufficient. However, when cells really start to lose control of their metabolism, it's often the mitochondria that send signals out to the rest of the cell to undergo apoptosis. If the mitochondrial DNA is mutated, they may not be able to send those signals. When the mitochondrial DNA is mutated, they lack the ability to send those apoptotic signals to the rest of the cell, and that can lead to potential tumors. So scientists are not only looking at the regular cancer-causing genes, these what we call proto-oncogenes, these genes that are involved in cell growth, and when they're doing their job normally, they're perfectly safe and in fact very necessary. But these proto-oncogenes, once they become mutated, can become oncogenes and cause tumors. But beyond that, as I've already mentioned, scientists are looking at the genetic information inside of mitochondria to see how that may lead to tumor formation. Beyond that is epigenetics. And this has to do not so much with the sequence information inside of our nucleus, the DNA there, or even in the mitochondria. This has to do with the three-dimensional structure of our DNA, which can be impacted by all kinds of environmental factors, of which I've highlighted a few in this picture. We've already mentioned diet. Diet is huge in changing the three-dimensional structure potentially of our DNA, not the sequence information, just the way that it's structured inside the nucleus. Not only that, but as I've already mentioned, high sugar diets tend to feed cancer cells. And one of the things that scientists are looking at that could be hugely important is when somebody already has tumor formation, reducing the amount of carbohydrates in the diet essentially may be able to starve tumors. But then there's other things like stress, cancer, exposure to chemicals. All of these things can have an impact on the epigenetics of our chromosomes. But the important thing to understand is while these things can have an impact, both positive and negative, on our chromosomes, one, these changes, these epigenetic changes, can potentially be passed down from parent to offspring. The other interesting thing with epigenetics is if you change behavior and you rid yourself of behaviors that cause problematic changes to epigenetic structure, the DNA can also change. That three-dimensional structure may also change. And so by having good habits, healthy habits, less stress, more sleep, better and more fulfilling exercise, those things, better diet, those can actually lead to positive changes in your epigenetics, while the bad things like smoking and drinking and drugs and things like that can lead to negative changes. But the epigenetic structure is potentially pliable. Why this is so important is this: the structure of our genetic material that allows for transcription of certain genes. And when certain genes that are supposed to be off get turned on, that may lead to cancer formation, just like when certain genes are supposed to be on, but the epigenetic structure changes and now we can't get access to those genes to make the proteins that are necessary. That can also be detrimental to a cell. Bottom line is there's a lot of things that have to go wrong for cancers to form. Things like an immune system that no longer is capable of recognizing sick and cancerous cells, poor diet, Genetic mutations, usually an accumulation of a lot of genetic mutations. Tumors have to be able to get excessive blood flow or increased blood flow to the area. A lot of times they have to lose their anchorage dependence and density dependent inhibition in order to form a tumor and then spread to other parts of the body. Cancerous cells also have to be able to ignore those very important checkpoints within the cell cycle. Everybody, so this was just a quick overview of how cancers may form and some of the things that we can do to hopefully prevent tumor formation in the future. As always, thank you so much for being here. We'll see you next week.